So we started with normal cell, and if we apply stress to a normal cell, first it will try to adapt. If the cell fails to adapt, it may undergo reversible cell injury, then irreversible cell injury. After this, the cell will undergo cell death. This cell death is having two patterns, morphological pattern. One is necrosis and one is apoptosis. And the most common cause, it was the hypoxia. Most common cause was hypoxia. So the two pattern of cell death, that is necrosis or your apoptosis. In case of necrosis, the first sign is cell swelling. But in case of apoptosis, this first sign is cell shrinkage. Cell shrinkage. This is the first difference at the level of first sign. Second is starting with the outside cell membrane. Cell membrane. Cell membrane in this case is disrupted. Here, cell membrane is intact. Because cell membrane is disrupted, the content may leak out. Content may leak out. Therefore, you will find inflammation. But cell membrane is intact. Nothing will leak out. So therefore, there is no inflammation. There is no inflammation in case of apoptosis. At the level of nucleus. There are three changes in necrosis. One is pycnosis, that is condensed form. One is karyorrhexis, and one is karyolysis. Karyolysis is just like a powder. Here, the nuclear changes are nothing but chromatin condensation. Only and only chromatin condensation. There is no karyorrhexis, there is no karyolysis, there is chromatin condensation. In this case, if we look at the DNA, this DNA breakdown in this case is random at any place. But here, DNA breakdown is not random, it is specific. Specific means at what position? That is, these are the nucleosome. The breakdown is in between two nucleosomes. So specific means internucleosome IN, in between nucleosome, internucleosome breakdown. And that is equal to 180 to 200 base pair. It is 180 to 200 base pair. So these are some of the small differences between two patterns of the cell death. First sign, swelling of the cell. Here is the shrinkage of the cell. Here cell membrane is disrupted. Here it is intact. It is disrupted, content leak out. so there may be inflammation. Intact, so no leakage of the content, no leak out, so no inflammation. Nuclear changes are 3. Nuclear changes only chromatin condensation. DNA breakdown is random, but it is specific. At what position? In between the nucleosome. And the fragment which are obtained, they are of 180 to 200 base pair in size. The next difference in necrosis and apoptosis is necrosis may occur in large group of cells, but here one cell or small group of cells, they undergo apoptosis. They undergo apoptosis. The next difference is it is always pathological. Always pathological. It may be physiological as well as pathological. Pathological. So with these differences, we are going to start our lecture on apoptosis. Apoptosis. If you break the term, ptosis means in surgery also we call drooping of the eyelids. Here also the term falling off means if this is a tree branch, it is having leaf. These leaves they start falling off. This is called as apoptosis. Falling off. This terminology, it was given by John Kerr. It was given by John Kerr. This was asked in ICAR 2021 exam. The term apoptosis was given by John Kerr. While working on one insect, C. elegans. So as the meaning of apoptosis is falling away of the cells from the body, this falling off, this apoptosis is regulated cell death because it is a pattern of cell death. It is regulated. It is also called as programmed cell death. In case of programmed cell death, apoptosis is one of the example. Other example are necroptosis and pyroptosis. They are also programmed cell death. One example is your topic, apoptosis. This process is energy dependent. So we call it active process. Apoptosis is an active process. The most important organelle involved is the mitochondria. It may be physiological or it may be pathological. It may be physiological or pathological. So first we'll see the type. Physiological apoptosis. Number 1, embryogenesis. Embryogenesis. Normally, this is the limb during embryogenesis. Because of apoptosis, it is converted into the fingers. It is converted into fingers. The space created is because of the apoptosis. So this is embryogenesis or organogenesis. The second example is the best example of physiological apoptosis is killing of neutrophil after completing their function. In case of inflammation, this is blood vessel and this is the neutrophil. Neutrophil comes out to the site where bacteria are present. This coming out is known as chemotaxis. So these are the neutrophils. They come out and they kill the bacteria. So neutrophil. The neutrophil will never enter into the blood vessel again. So what happens to the neutrophil? It will undergo apoptosis. That is, this cell will die. This neutrophil will die. So once their role is completed, they may undergo apoptosis. So chemotaxis is always unilateral. It is never bilateral. It is always unilateral. So they will kill and after they are also dying. By which process? Apoptosis. So this is killing of the neutrophils once their role is completed after the phagocytosis. Number 3 is the elimination or removal of self-reactive or autoreactive B cell or T cell. Why there is elimination? Why elimination? So the reason is to prevent autoimmune disorder, autoimmune diseases. These self-reactive cells, they are responsible for autoimmunity. They are responsible for autoimmune diseases. So to prevent occurrence of autoimmune disorder, we destroy this self-reactive cell by the process of apoptosis. If we do not destroy, it may lead to autoimmune disorder. So these are destroyed by the apoptosis. Next is pathological apoptosis. Number 1, viral hepatitis. There is something called concealment bodies, which are nothing but apoptotic bodies. These are present. That is pathological. Second is mild irritant. Mild irritant may lead to apoptosis. That mild irritant may be hypoxia. That may be thermal injury. And that may be radiation injury. That may be radiation injury. But mild irritant that may lead to apoptosis. Next topic is the morphological appearance of apoptosis. Number one, cell size. Cell size always decreases, so we called cell shrinkage. Cell shrinkage. This is the first sign. This is the first sign. But if you remember, most characteristic feature of apoptosis, that is your chromatin condensation. Why it is most characteristic, I will explain at the end of the lecture. But remember, this is the most characteristic features, chromatin condensation. Third is there is no inflammation in case of apoptosis. Why? Because cell membrane is intact. There is no leakage out of the enzymes and other material. Number fourth is cytoplasm. If you look at the apoptotic cell, this nucleus is chromatin condensation, and this is the cytoplasm. This is more red or pink in color, more eosinophilic. It is more eosinophilic. Number fifth, morphology of apoptosis. Number 5. If we compare normal cell versus apoptotic cell, then this is the cell membrane having lipid bilayer. So all lipid, they are facing toward the cytoplasm. But in this case, the lipids are facing away from the cytoplasm. They are facing away from the cytoplasm. Some lipids are flipping outward. They flip outward. The name of this lipid is phosphatidylserine. This was asked in ICAR 2018. ICAR 2018. Which molecules exposes on the surface of apoptotic cell? Phosphatidylserine. In case of a normal cell, these are facing toward the cytoplasm. But in this case, away from the cytoplasm or outside, on the outside. flip outward, phosphatidylserine. There are phosphatidylserine and one more molecule, that is thrombos thrombospondane thrombospondane, that also express on the apoptotic cell. Thrombospondane. The question is how to diagnose apoptotic cell from normal cell. So if you have some dye which loves lipid, for example, this is the cell of apoptotic, where this is the phosphatidylserine. This is also phosphatidylserine. If we have some dye, lipid dye, which combine with this dye is known as annexin V. This is known as annexin V. This annexin V, it will bind with the phosphatidylserine. So this is apoptotic cell which is having phosphatidylserine outside. So this dye, this annexin V, it will bind this molecule. So this lipid dye, annexin V, is help to diagnose apoptotic cell from the normal cell. So if you have a group of cells, suppose this is the normal cell having phosphatidylserine inside, and this is the apoptotic cell which is having phosphatidylserine outside. So the annexin V molecule, it will bind with these two. It will not bind with the normal cell because they are inside. It will bind with the outside molecule, annexin V. So this will help to diagnose. So next morphological point is the bleb formation. Is the formation of bleb. Why they are forming? What is the reason behind this? They are forming because of the cytoskeletal protein, because of the cytoskeletal protein deformities. This is a cell which is having cytoskeleton. If cytoskeleton damage, then it will form bleb. It will form bleb. This is known as bleb. It is having cytoplasm, some organelles. So there are multiple bleb formed in the cell. When this bleb get detached, it is known as apoptotic body. Apoptotic body. It is composed of cell membrane. It is having some cell organelle. It is having cytosol, and plus and minus may or may not have nuclear fragment. May or may not have nuclear fragment. So plus minus nuclear fragment. That is the composition of apoptotic body. What is the example of apoptotic body? This is the Councilman body. Where it is found? It is found in viral hepatitis inside the hepatocytes. So if this apoptotic body is formed, what is the fate of apoptotic body? What is the fate of apoptotic body? This apoptotic body is also having phosphatidylserine on the surface, PS, phosphatidylserine. These are some macrophages. Macrophages. Macrophages have receptors and they will eat this apoptotic body. They will eat. So the fate of apoptotic body is phagocytosis. Fate is they will eat, phagocytosis. Next is mechanism of apoptosis. What is the basic mechanism of apoptosis? So just remember two words. One is initiation. One is initiation. And second is the execution. Initiation may be through intrinsic pathway or through the extrinsic pathway. That may lead to execution. From the execution, there will be activation of endonuclease. And these endonucleases, they may lead to chromatin condensation. Chromatin condensation. So this is the most characteristic feature of apoptosis. Most characteristic feature, because in intrinsic pathway and extrinsic pathway, in both pathways, the end result will be the same. That is your chromatin condensation. So whether it is intrinsic or extrinsic, end result will be chromatin condensation. So this is hallmark or most characteristic feature. So first we'll start with the before going to mechanism, we'll see there is one term caspase. So caspase are C for cysteine. So they are cysteine containing As. As means cysteine containing protease. Acting at ASP. ASP means aspartic acid residue. Aspartic acid residues. So if this is a protein chain and this is the aspartic acid residue, they will act at this place. So these are the cysteine containing proteases which are acting at the level of aspartic acid residue and remove that piece away. So these are caspases, enzyme. So these caspases, if you remember, in case of intrinsic and in case of extrinsic pathway. Extrinsic E for 8, EIGST, means caspase number 8. And in case of intrinsic, it is number 9. N-I-N-E. In the name itself, I-N. So, I-N, 9, number 9. The intrinsic pathway role is number 9, extrinsic pathway 8. This 8 is for the animals. In case of human being, it is easy to remember 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 letters in human being. So number 10 caspase in case of human being. So for extrinsic pathway, remember 8 or 10. For intrinsic pathway, only remember 9. And what are caspases? They are acting at aspartic acid residue. Simply ases means they are enzymes. They are cutting at aspartic acid residue. Because apoptosis is programmed cell death, so there is role of genes. There is role of genes. These genes are of two types. One which promote. They will promote apoptosis. So we call them pro-apoptotic gene. And one is against them, so anti-apoptotic genes. So what are the pro-apoptotic genes? They are Beck's gene, BAK, Beck gene, Beck's Beck, then BCLXS. S means stimulate apoptosis. Then the most important is BH3 only protein family. Protein family. This is a family, so the members are Puma, BIM, and BID. These are the members of BH3 only protein family. They are pro-apoptotic gene. They promote apoptosis. Out of this BH3 family, they are known as stress sensor of a cell. Stress sensor. They sense the stress, if the cell is in stress or not. Anti-apoptotic gene. They are very important is BCL2 gene. Next is BCLXL. L means it lowers the apoptosis. And MCL1. These are the genes which play a role in your apoptosis. So, the basic mechanism, as I told, it is initiation, then execution, then endonuclease activation, and then chromatin condensation. This initiation is involving intrinsic pathway or extrinsic pathway. Here, caspase number 9 and E for caspase number 8. In human, it is 10. They are playing role. Execution role is played by caspase number 6, 3, 6, 7, and 12. 3, 6, 7, 12. They activate then chromatin condensation. So first we'll see what is intrinsic pathway, then we'll see what is extrinsic pathway, and what are execution cascades. So first is the intrinsic pathway. That is also called as mitochondrial pathway because there is role of mitochondria. It is also called as apoptosis pathway because there is formation of aptosome complex. So for this pathway, what we need is the signal. That signal will come because it's intrinsic. Intrinsic means signal will come from inside the body. Suppose that signal may be number one, deficiency of growth factor, deficiency of hormones, growth factor. No growth factor, no growth of the cell. Second, signal may be cell stress. Signal may be any kind of cell stress. As this is the cell, and on the surface of cell we have BH3 family protein, BH3. They will sense the stress. They will sense this BH3. The example of BH3 is the puma, PUMA. So these are the signals. How this signal work? Before that, we'll see the structure of a cell. Structure of a simple cell. The surface is having BH3 sensor. They will sense the stress. Inside we have mitochondria. Mitochondria. This is outer membrane. And this is the inner membrane. And here we have cytochrome C. This inner mitochondrial membrane have backs and back protein. Backs and back protein. This is outer, no? This is inner one. And this outer mitochondrial membrane is having which protein? BCL2. It is having BCL2. In the cytoplasm, there are presence of IAP, inhibitors of the apoptosis, IAP. So these are molecules present in on the surface BH3, inner mitochondrial membrane, backs and back, outer membrane BCL2, in the cytoplasm IAP molecule. So with the intrinsic pathway, what is the signal to start a pathway? That is a stress to the cell. If the stress comes, who will sense BH3 family protein? The example is puma will sense. Puma will sense this stress. This BH3, as we know, this is the mitochondria, outer or inner membrane. This BH3, it will stimulate backs and back. It will inhibit BCL2 protein. BCL2 in the outer membrane, or backs and back they are in the inner membrane. So this BH3 puma will stimulate this protein and inhibit this protein. What is backs and back? They are product of pro-apoptotic genes. So they are pro-apoptotic. It means they will promote the apoptosis, and this was anti-apoptotic. So you have inhibited anti-apoptotic. It means you are stimulating apoptosis. So back and back stimulated, it means pro-apoptotic product is stimulated. If you stimulate these two, they will form oligomer. Oligomerization will be there. Oligomerization. And they will form some mitochondria outer membrane permeabilization. That is nothing but some of the pore in the outer membrane. So what happens in intrinsic pathway? There is one signal, and that signal is stress. That stress is sensed by the BH3 protein puma that is present on the surface of the cell. This puma will stimulate backs and back. It will inhibit BCL2. These backs and back in the outer membrane, they will come to the outer membrane and they will form some of the pore, MOMP, outer membrane pore. From these pores, say inside, in between two membranes, cytochrome C is present. This cytochrome C will come out. Here, one more molecule, SMAC. It will also come out. SMAC. So these molecules will come out in the cytosol. From where? From intermembranous space. In between two membranes, inner and outer. From the intermembranous space, cytochrome C will come out. SMAC will come out in the cytoplasm. Now what happens? So from the mitochondria, this cytochrome C and SMAC both are coming out. This cytochrome C will combine with one molecule APAF1 and form the complex apoptosome. Apoptosome. Apoptosome is nothing but combination of cytochrome C with APAF. This will help to convert procaspase because the pathway is IN means 9, IN, that is 9. So procaspase 9 into caspase 9. It will convert inactive into active. Who will convert apoptosis? In the cytoplasm, there are some protein IAP. They are inhibitor of apoptosis. They are present. They will inhibit this formation. Once this SMAC comes out, this will inhibit this. It means what? It means this SMAC will favor apoptosis. It is pro-apoptotic. Where it was present? In the mitochondria. Now it is in cytosol. So it is pro-apoptotic. So this caspase 9, they are the initiator caspases. This will activate caspase 3, 6, 7, and that will activate endonuclease. Endonuclease. And that will lead to chromatin condensation. Chromatin condensation, the most characteristic feature of your apoptosis.
Hello, clear the numbers. Hello, 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 hello.